Glucose is as a type of carbon molecule.
Sugar.
Okay, it is a sugar. And does anybody know, kind of, let's go on a little bit more broad term of what type of category sugar might be in? Football, you guys, football players, they usually eat this. It gives you a lot of energy right before a game.
Protein.
No.
That's how you exercise.
Starch, which is what? Bigger picture, bigger name.
Carbs?
Carbs, yes. Carbohydrates. Okay? This is a part of a carbohydrate. Shh. You digest them. And sometimes, but the thing is that in your digestive system, you have these reactions. Sometimes you need a jump start in order for it to go a little bit faster so you do not consume as much energy. This is where a catalyst comes in. A catalyst will speed up a reaction. And I put RXN for reaction because I start to write in code. Okay. Catalysts will speed up reactions. So you have carbohydrates that are going to fuel your body with, let's say, a certain game. Carbohydrates break down faster. It gives you that burst of energy. This is why it's important to have breakfast in the morning. Carbohydrates are very good to start with. However, it usually burns off, and this is why you're eating during second block on many occasions. Football games, you eat a huge carbohydrate meal. I can say that with too. Soccer, you eat a huge carbohydrate meal in order to have that energy. You don't eat protein. It will make you throw up on the field. Because protein is very heavy and it digests less. I had, during my first years of teaching, I taught at a very huge football private school, and the football coaches would give the players a steak meal 2 hours before the game. And they would come tell me, they're like, oh yeah, that's what you need before a game. I'm like, are you kidding me? To have a steak? Eat a steak and then go running. Could you think about what's in this right here? You'd be like, in your mask. My husband is a soccer coach for college. He went to a tournament. He noticed that he was playing the team that was next to him. They went to lunch. Him and his team were having like either spaghetti with marinara sauce or salad. No like thick dressing and all that stuff. The other team he saw was eating, was it Alfredo, all that cream and chicken and just that fatty food and heavy food. They were very slow on the field because you don't want to eat a very, very heavy meal that consists of high in proteins, high in fats, because they're last to digest. Anyway, why am I telling you this? Number one, health reason, but number two is that in order to speed up these processes, you use a catalyst. And all enzymes are catalysts. They speed up reactions. A good example is papain. This is in, what is it called? Mangoes. Okay. Mangoes have this enzyme, and it's called papain, and it actually speeds up the digestive system or the digestive of protein. So let's say if I do this in a chemical reaction, if a chemical reaction goes like this, okay, so it starts right here. We're going to talk about this more later. This right here is the energy to get it started, E-N-E-R-G-Y. That's fine, but now you enter this pepsin or this papain. Now, I'll put this in green. This is the enzyme. Now we just speeded up the reaction so we're not using as much energy. In physical science, I think last year or maybe in a different type of science, maybe you guys talked about potential energy? You're like,
yes,
yeah, maybe. So have you heard of it before then?
Yeah.
Does anybody recall what it is?
No.
So let's break down the terms. We all know what energy is. What does potential mean? In your own words, yes.
Oh. Never mind.
Okay, someone want to help Michael?
It has like the capability to do it.
Good, the capability of doing something. Good, that's exactly what it is. So in reactions, we have this potential energy. We want it to have the capability to do work, which will then turn into kinetic energy, the actual work doing.
Actually, it's moving, right?
That's when it's moving. Excellent. You guys have great memory. That's very cool. So enzymes will decrease that energy required for it to work. So it just shortens it. Okay. in your lab, that's what you guys are going to be testing is the effects of certain things on enzymes. There are many things or factors that can influence the rate. What was the title of the lab? Does anybody recall? Yeah, I got to just look at the lab.
Enzymes?
Okay, enzymes, but what was the purpose? pH. So then what are you going to be looking at, Paige? Okay, the acid levels, but you're looking at the rate of pH on enzymes. So what does that mean? Who can repeat back to me what I just said in their own terms? So your title is, or your purpose of your lab, is the rate of pH on enzymes. What did I just say? The rate of pH on enzymes. So what does that mean? The effect of pH on enzymes. So pH will affect it. You guys are going to be looking at different ranges of pH, the effect on the enzyme. There are a couple other ones from your notes that can affect. Can anybody go back to their notes and tell me what are some other things that can affect enzymes reactions? Tariq, you want to look? Do you have your guided notes? So we have pH. What can affect it? Is it affect or effect? Effect. So she's thinking. So we have pH. What's some other things that can influence? Look it up. Go on Instagram. They're on your notes. Your guided notes. Look on the PowerPoint. What are some things that can regulate it? Temperature. Very good. And shape. Excellent. Okay, that's a good point I want to talk about right now is shape. What I'm drawing here is a substrate complex. Okay, right here is my enzymes. I can't write. Lock and key. Enzymes have what's called a lock and key method. What do I mean by lock and key? Tariq, what do I mean by lock and key? It's a lock and a key. Does any key open any lock?
No.
So what do I mean by lock and key?
Be quiet.
What do I mean by lock and key? If you just told me that, hold on, I want Tariq to answer because I'm talking to him. If you just told me not every key will open every lock, what do I mean by lock and key method? What do you think?
Not every key will open every lock.
Okay, not every key will open every lock. So then there is a what type of key will open what type of lock?
Specific key?
A specific.
Yeah, specific. Okay. Enzymes are highly specific. Not every enzyme will open or will combine with a substrate complex. There is one complex for one enzyme. So in this case, here's my substrate complex. What type of key, what type of enzyme is going to fit? I have a circle, a triangle, or a cloudy triangle. Okay.
What's that thing called? The
Substrate complex. And what happens is that this combines in order for the enzyme to work. It produces a product, speeds up a reaction, and then these two disjoin or come apart again for it to do somewhere else. Now sometimes with this thing, shape can influence. Something can attach to this. Let's say I put like a little happy face type of thing. I'm just kind of drawing funky little shapes here. This attaches this, and now this just turned into this. Can this fit?
No.
No. So shape can affect it. Sometimes something can attach to it, and it's called an inhibitor. It can either change the shape, or it can say, no, you can't go. So it can do one or two things. An inhibitor can change the shape so it can't fit anyway, or this inhibitor can say, no, you can't go. It's like a go, stop, Simon Says type of thing. So that's how shape can influence. How temperature can influence is sometimes you have your rate of your reaction, so it goes uphill, it hits a certain temperature, the enzyme just won't work anymore. So it stops. So there's like a prime temperature, like Optimus Prime.
Optimus Prime.
Optimus Prime. So these are the enzymes, and this is what you guys are going to be doing on Tuesday. You're looking at natural enzymes. There is a natural enzymes in potatoes and turnips. You're going to look at both of them, and you're going to see the rate how pH affects how things move. And you're taking a disc of paper, and you're going to be putting them in your test tubes. Okay. So let's draw. In block 1, there was some confusion of exactly what you were doing. You're going to be having your test tubes, and here are your test tubes. I'm almost done. So here are some examples of test tubes, and you're going to have a range of pHs. So one's 4, 5, 6, etc. You're going to be putting, quiet please, you're going to be putting 3% of H2O2. Does anybody know what this is? You have it at your home. Put it on your ears, put it on cuts. You can put it in, your... hydrogen peroxide. Okay, hydrogen peroxide. This key, you guys, highlight this and note this. This has to be exact when you put that in there. If it's not exact of 1 mil, because it's going to be 1 mil, it can throw your whole thing off. So you have to be so precise, and you get a micropipette, so you have to look at it. There is no eyeballing. There is no, it's kind of there, it's kind of at 1 mil. But then you are taking your disc, it's a hole puncher, a hole puncher, and you're taking these discs. You're going to put them in your potato solution that I'm going to have up front. Tweezers, put it inside, soak it up a bit. Then you put that little paper disc in there, and you're going to time it from the time it goes down to the time it goes back up at each pH. That's all you're doing. So you're doing this in potatoes and then turnips for each and every single pH from 4 to I think 11. That's it. But I think students were getting confused because they have to put this in here and then they put this in here. It's time consuming to set up. But once you get going, you can get going. Okay. So I just wanted to clear. If you guys have questions again on Tuesday, by all means, please ask. So that's it for the lecture for today on enzymes. Your homework is to just watch a small lecture. I haven't made it yet. It should be up hopefully tonight. If I mess up at anything, I'll just put the PowerPoint back up and tell you what slides. And so it's going to be on chemical reactions and organic molecules. That's all you have to do for Tuesday. Tuesday is going to be the lab. I'm hoping to get another food lab in, and then Friday will be your test.
Can we make ice cream again?